Hello, everyone. Many thanks for being here today. We are proud to have Professor Mehmet Toner with us. Professor Toner is a professor of health sciences and technology at the Harvard MIT Division of HSD. He serves as a member of the senior scientific staff at the Shrines Hospital for Children. He's also a co-founder of the Center for Engineering in Medicine and founder of the NIH BioMicroElectroMechanical Systems Resource Center at the Mass General. He's also the director of the Biomedical Engineering Research and Education Program for Physicians at Mass General. He's a member of many national and international professional committees and serves on the editorial board of many scientific journals. He has served on many national and international panels. In 1994, he was recognized by the Y.C. Fung Faculty Award in Bioengineering from the American Society of Mechanical Engineers. In 1995, he received the Whitaker Foundation Special Opportunity Award. In 1997, he won the John F. and Virginia B. Taplin Faculty Fellow Award given by Harvard and MIT. Later, he was selected to become a fellow of the American Institute of Medical and Biological Engineers, where he is serving. He has published over 200 scientific publications and delivered over 350 invited and scientific meetings. We are privileged to have him here today. Dr. Tanner, the floor is yours.
Thank you, Patricia, for the invitation. And when you gave that presentation a couple of months ago to ATP Bio, which is a center we have jointly with John Bischoff and colleagues at Minnesota on biopreservation, it's an NSF ERC center. You mentioned that cells could be sensors. And so I've never looked at it that way, but it resulted in this invitation. What I would like to do is just talk about circulating tumor cells. And for those of you who don't know much about them, is cancer kills by spreading across the body. It's called metastasis. And these cells are transit in blood. They're extremely rare, one in maybe 100, 200 billion blood cells. And in a sense, they're a sensor. You can sense cancer very early. You can monitor it very accurately because these are the cells that ultimately spread and cost lives. And then remember, 9 out of 10 deaths in cancer happens because the cancer spreads. Only a few cases, glioblastoma, brain cancer, and a few others, that cancer, local cancer, primary tumor can be deadly. And we have been working in this area for over 20 years. And what I would like to do is just at a high level share that journey, why we do it, and what's the motivation, and what kind of information we can get, and hopefully that will catalyze some interest. Let me try to share the slides. Are we in the?
Yes.
Okay, perfect. And let me get these things out of my view so I can see my own slides. Okay, great. And so it's really, this is the story of developing technologies for isolate rare circulating tumor cells called CTCs. And just the usual disclosure. Okay, where's this slide not moving to the next slide? Somehow my slides are, oh, here we go. Sorry.
It's not moving.
Yep, now it should work.
Ah, yes.
Okay. And the banner was closing the arrow. Anyway, in the form of disclosure, then we have a number of companies, but these are all conflict issues managed by Mass General Hospital and Partners Healthcare in accordance with their conflict of interest policies. And this field is not new. It always seems to me that everything that we think is done sometime in 1800s. The first paper that we know, or I know, is 1869 by Ashworth and resident at Monash University, published a two-page article in the Australian Medical Journal. And here you see the primary tumor. This is hand-drawn from the eyepiece, the microscope. These staining techniques, a lot of them actually developed in this paper. And you see a tumor cell. This is the primary tumor. You see the nucleoli, and this is the blood. This was post-mortem. The patient was a young male patient that passed, and the sample was taken from the vein. And you see 3 tumor cells in circulation. And it really, this paper said the first time that identical of those cells in the primary tumor, it's in the blood, may tend to throw some light upon the mode of origin of multiple tumors existing in the same person. So it's really a brilliant study, a two-page study. And in 1960s, there was a textbook written on circulating tumor cells, and you see Dr. Monash Ashworth here in the background. And the bottom line is these cells, now we know that intravasate from the primary tumor as soon as the tumor is the head of a pin containing a few thousand to tens of thousands of cells. And they're typically in a very metastatic patient, 1 in a billion blood cells is a CTC. Typically, it's 100 to 200 billion cells. There's one circulating tumor cell, and they extravasate and form metastatic cascade. Just to put it into perspective a little bit, there are 37 trillion cells in the human body. Total number of mammalian cells. Of those, 85% are blood cells. In a sense, we are all blood cells walking. It's 75+% of all the cells in human body, mammalian cells, are blood cells. So blood is an amazingly powerful source for all kinds of diagnostics. And we really are tapping only the tip of this resource. Mainly because we can't read these extremely rare cells that control a lot of processes, from prenatal diagnosis to stem cells to progenitor cells to controlling a number of pathological processes such as heterotypic ossification, even on issues related to stroke and concussion, where rare cells leak into circulation that has tremendous potential. In the case of cancer, applications involve if the new targeted therapies, is it the right mutation, right therapy, immunotherapy? They're very toxic, very expensive. You don't want to continue if it's not working. Is it working? So understanding predictive as well as early response. Drug resistance emerges in almost in every single therapy within the year or several years, and monitoring that and realizing it early and changing treatment to second and third line drugs, which are many more now. And early detection, of course, is the holy grail. And personalized oncology, if we can find these cells, can we grow them and test for that patient drugs and see which drugs will kill the patient's own tumor cells and recurrence detection. So the applications are significant, and it really gives from a sensor's perspective. I really love looking at these cells as sensor. I have not didn't think that way until I met Patricia, and it really provides a real-time window into a very complex biological process, cancer, that evolves. It's very dynamic. It's an evolving disease with the pressure of treatment and its own spontaneous reasons. It keeps changing. And so ability to use these cells as a sensor to monitor cancer is quite exciting, I believe. And this slide is just to provide a sense of the technology. And I'll show you how we use microfluidics to isolate these cells. And microfluidics is a field that started in 1980s and with the aim of processing tiny amounts of fluid or individual cells with an incredible sensitivity of single particle and driven by techniques called miniaturization, microelectromechanical system, integrated circuits, microsensors, microvalves, things that are very familiar to IEEE members who are in the audience. But what we did is that we went the other way around. For clinical samples, if you want to detect a rare cell and in 200 billion cells, that's going to be 2 tubes of blood. A single tube has 50 billion cells. A finger prick has 500 million cells. A pint of blood has 3 trillion cells. And the total human body, as I mentioned, has 37 trillion cells. Today we have techniques that we can actually process the entire 5 liters of blood for various applications, not for rare cells, but for other applications. So we've pushed microfluidics to a point where I call it we hook a garden hose to a microfluidic chip and do that in this with a single cell precision. And I want to show you how we evolved the journey in circulating tumor cells. By the way, if you have any questions, please put it in the chat or just stop me. And Patricia, if there's a question in the chat, I may not be able to see the chat in real time. Just let me know, and I'll be happy to answer the questions as we go along. And we have built our own clean room at the hospital. It was a unique facility at the time to be able to do these innovations faster and cycle time of innovating and changing designs and testing different things. So it's within the biological laboratory that we have our own small clean room. And then when things get more complicated, we go to Harvard, MIT clean rooms that are much more bigger and established, as well as we go to foundries and outside sources. And you'll see some of those examples. And so, for example, this is the first paper we published that used microfluidics for handling both very complex bodily fluids, not in a buffer solution with several cells, but this is whole blood and with billions of cells. And the initial technology to isolate these cells, most cancers are epithelial cells. They have a protein on their surface called epithelial cell adhesion molecule. So we created this microfilter type system that we coated these posts with an antibody that binds to EpCAM, epithelial cell surface molecule, and push the blood, whole blood through. I won't go into really nice fluid mechanics of how we distributed these posts, but these posts are about 100 micron tall and 20, 30 micron wide. It's done with deep reactive ion etching at MIT. You see it's in the silicon here process. It's about the size of a glass slide or half of a credit card. And about 100,000 of these posts, the blood goes through. And if it has the right surface epitope, it binds to the post. And you see here in prostate cancer, you see the post, you see the cells. Cells are about 10 micron. These posts were a little bit larger, about 45, 50 micron. And you see the pink is that the cell is actually dividing. So not only we can get the cell, we can get molecular information, whether it's dying or it is dividing. And here you see blue, just the nucleus is blue. That's another one here. With green is the tumor cell, blue is the nucleus. So this cell is actually a blood cell and doesn't stain for EpCAM. And after 7 years of work that we have, I won't go into any of the details of this technology because I will abandon it quickly. We have figured out that the CTCs are very heterogeneous and dynamic. So each cancer, each stage, we needed to design the right markers and right ways of isolating. You can see some of these CTCs are covered with platelet, you can't find them because there's no surface EpCAM to bind. It's covered. Many of them are going through what's called epithelial mesenchymal transition. So they're shedding their marker, in this case, EpCAM that we use. So it became very complicated to do these individualized conditions to optimize. And then we said, can we actually do agnostic to the cancer type? Can we develop a technology if it is a needle in a haystack, instead of like we are doing in the prior slide, trying to find the needle, can we get rid of the haystack, which is the normal blood cells, to find the needle? We call it negative depletion. In other words, you are getting rid of blood cells to find the tumor cells. Then I don't care which cancer, all tumors can be found, tumor cells in circulation with an approach like this. Given that in 2 tubes of blood you have in 100 billion blood cells, to find one cell, you need to get rid of 99,999,999,999 cells without losing that one cell. That is very challenging unless you do, we believe, microfluidics and single cell resolution. And to do that, we have developed a technology. The crux of the technology comes from a discovery we made in 2007 called inertial focusing in these microchannels. And just bear with me to go through this, that hopefully will make sense. So these are UC cells. When you're on laminar flow, if you come to a channel, a cell that comes here has to come out exactly in that same location. It won't change its streamline because the flow is in laminar flow. And we will increase the flow rate here in time, and you'll see that they will start focusing, which according to laminar flow, they shouldn't. And you see it comes here and it comes out exactly in that location. We keep increasing the flow rate. You might see some already, you see now they're almost in 2 locations in this geometry, and they're also equidistant from each other. You see they're in these 2 locations in the channel. And what happens is, if the particle size is at the same length scale as the channel, the particle feels the shear stress pushing it towards the wall, and the flow around the cells pushes it away from the wall, and they find this perfect location, and they are equidistant from each other. And it's extremely high throughput. We can push these things at 130 meters per second. That will be faster than any Formula One car race top speed today. Or it will be equivalent of about these cells moving at 130 meters per second. If in a tennis, the top serve is 160 miles per hour, and that will take about 200 milliseconds. Here, you will move in the same amount in less than millisecond. So it is so fast, but also shear stress is very low, and it's high throughput. It's extremely precise, and we can also change the channel geometry, what we call wiggly, to bring, for those of you who are fluid mechanics people, Dean force to create vortices, so that you see at the inlet, the particles come in any location, but at the outlet, they are single file. And so we can do this very fancy ordering. We can parallelize it, and we can separate particles. So with that in mind, the way we will use this is, if I go back to, we are going to put if these are the white blood cells, not red blood cells, not platelet, white blood cells, and not tumor cells. We know their surface antigen, so we can bind magnetic antibodies to white blood cells. Because they are lined up in a single file, we can actually use magnetic field to deflect them. So here is a white blood cell with magnetic beads attached to them. We just in the tube, in the whole blood, we mix it 2 minutes. We have protocols, and push it through a schematic of a conceptualized microchip. the first part of the chip separates things in size. Red blood cells and platelets are much smaller than nucleated cells. So this portion with these posts that we do, it's called deterministic lateral displacement, and they are shifted, and then the white blood cells are separated. So that's the first part of the chip. And then it comes to the inertial focusing region, the concept that I showed you that's fundamentally important for this innovation, inertial focusing region, where they become into a single file with green tumor cells mixed amongst the white blood cells. And we put a magnetic field here, shift the white blood cells and collect CTCs here. In the CTCs or cells spend about a second in the chip, so they come from blood and they are separated. It's continuous flow, extremely efficient. The early stages of the technology where we had this portion in silicon etched, it's about $1,000 to make that chip. And the other portion, inertial focus and magnetic portion here in the metal, and that we can make using lab prototypes called elastomers. But we have scaled this up to a plastic chip, working with a company called Stratec in Germany. And it's the good old Blu-ray technology. They got out of business, so we are making microfluidic chips now. And blood comes here. This chip can process about 20 ml blood in half an hour, and it has a resolution of sorting 100 to 200 million cells per second. This is what we call the pachinko device or size-based sorting. Blood, there are 128. This doesn't spin. We just use the CD, so it doesn't play music. It doesn't spin. We use the CD technology. We didn't want to change the form factor. That would have been millions of dollars. We printed our chip onto this plastic, but it's two parallel chips to get the higher throughput, so it's symmetrical. And there are 128 channels here that can deflect red bloods and platelets, and then the chip comes and it's inertially focused here, and then we do it twice. First, we get the cells that have a lot of beads on them, and then we go to a higher magnetic field area where you get the cells that have lower number of beads. And then we collect the tumor cells from this hole, and then red blood cells from this hole, and white blood cells from this. And this chip has actually a third layer that controls all the inlets and outlets, and a processor that we have also developed that a technical assistant can put the blood, mix with the beads, push a button, and you collect your tumor cells here. And so now we are doing negative depletion, getting rid of red blood cells, platelets, and white blood cells, and you get highly enriched tumor cells and a few other cells that come along. And this is after, since 1869, after 150 years later. And in melanoma doesn't have any marker, so you couldn't positively find these cells. And you see the melanoma from fine needle aspirate. That's the primary tumor, like Ashworth did 150 plus years ago. And you see the nucleus, you see the nucleoli, that's a tumor cell, melanoma cell. And here in the circulation that we captured with our technology, just to show you that because the cells spend less than a second on the chip, they're not stained, they're not anything. So they're in pristine live cells that can then be used for high quality diagnostic imaging. But our goal is not so much imaging as a sensor. We want to use them as a molecular sensor to get every single possible molecular information from these cells. I've showed the a number of different papers here. I'm not going to go into details, but just to give you the richness of different studies you could do if you have the cells that end up killing people in a tube, and then you can start sequencing them, culturing them, and look at mutations. So I'll give you 4 or 5 different examples to let me see. There are some questions. I'll answer them. I just saw it in the chat. Is the speed of fluid just to perform a scan very quickly? Speed of the fluid is the process, the volume that we need to handle, 2 tubes of, but we do not do the scanning in real time. The analysis is done later. In cancer patients, you really don't need that, like infectious patients. Can you identify cells based on where they are in the channel? No, we want to do the molecular analysis. Just it's not the cell counting that's useful to a certain level, but it's not that critical in today's management of cancer patients. We really need that molecular information. You'll see that in the next few slides. And but the point here is that because they're untouched, undamaged, the RNA is intact, cells are intact, so we can get some very high quality information from these cells. And so, for example, one of the early studies we did is targeted therapy. If a patient in lung cancer has EGFR mutation, there are drugs specifically for that. If there are other ALK translocations or KRAS, there are drugs for this. So we would like to know that so we could give the right drug to the right patient at the right time. And in this one, we find the circulating tumor cells, look for EGFR mutation, deletion, and the patient was put on the drug, gefitinib, responded very well. At some point, the patient stopped responding, and this patient later passed away. But that's when the resistance gene T790M was expressed. So we could find that too. So we could not only say, okay, this is the drug you should give, and then you monitor it continuously. And at some point, if the resistant gene is expressed, you go to the alternative therapy. So it shows you how you can monitor these targeted therapies. Again, could be toxic, expensive, and you don't want to use it when it stops being beneficial to the patient. You don't want to delay the second, third line treatments. We can also look for more basic perspective discovery of new biomarkers. Are these cells that are circulating, we know many of them are dying, but some of them can survive. Are there specific markers that make them survive? If so, could those be target for therapeutics? And we have done sequencing of the circulating tumor cells as well as the primary tumor blood and ascites to see what is expressed in circulating cells is unique. And one of the things that is a signaling pathway in the cascade, Wnt2 signal was extremely high. And we also looked at these in patients, not just in animal models. We find it in animal model. So that could be a potential target for targeting the circulating tumor cells and specifically killing them. We have also looked at very common use now, immune checkpoint therapy. It's almost every cancer patient at some point is going to get immunotherapy. And especially melanoma, it's quite effective. We have created the 19 gene panel to find these circulating tumor cells and then sequence with droplet PCR and create a CTC score for the patient longitudinally. So if you look at CTC score, which is the expression of these 19 genes, and it's a mathematical equation that includes these genes. It doesn't have to have all of them. And if you do CTC score like you do in clinical medicine, you don't really see a difference between those patients that do better versus that do worse. But because we can do multiple measurements and measure like a good engineer a delta CTC score, suddenly these signals, the noise is much less. The signals separate from each other, and you can see those who are going to survive versus those who are not going to do well. So you can really monitor these patients very closely and have a prognostic information. And so just going through a number of examples. Another one that is extremely important, cancer becomes deadly when it becomes epithelial shifts to a mesenchymal phenotype. This happens during the process of evolution, and but it's never been shown before in animal models. They've been shown, but not in actual patients. So we collected CTCs from breast cancer patients and looked at the EMT transition, epithelial to mesenchymal transition, which is important. And so we labeled the CTCs with epithelial multiple markers in a single color and multiple markers mesenchymal. Here you see in the same patient, some cells are completely epithelial, some cells are completely mesenchymal, some are in mixed phenotype. So you can see the dynamics of this just from this figure, and these are from patients. And so what we did is also we looked at the tumor embolus. There is also older theory about the mechanism of metastasis, not related to epithelial mesenchymal transition, but there's a belief that there are tumor clusters or embolus that is leaked from the primary tumor. It gets clogged into one of the vasculatures, and then it grows into the tissue. And so that's a different mechanism. So now we can probe these two mechanisms in the same patient. So when we looked longitudinally to one of these patients, I call it index patient, you see that the patient had a lot of circulating tumor cells, pretty advanced cancer, and they were all epithelial. Blue in this graph is epithelial. And as the treatment successfully lowered the number of cells, the phenotype became much more epithelial, so less aggressive. And then the patient stopped responding. You see, again, it becomes much more aggressive epithelial phenotype. A new drug is given, second-line drug. The patient responds beautifully for a few months, but again, the phenotype goes very mesenchymal, very aggressive, and the patient passed away. And but interesting, if you look at this one, you could say, oh, epithelial mesenchymal transition is driving the metastatic process. To our very surprise, when you look at for circulating tumor clusters, we have seen, which you don't expect seeing clusters when it's mesenchymal type, but we actually saw clusters more in the mesenchymal form. So these two phenomena was not completely decoupled from each. The cluster is very important. Here you see a beautiful cluster. The green is the tumor cell, red is the blood cells that are within the cluster. This is a relatively large cluster. Many of them are 2 to 4 cell clusters. But we see clusters that could be 50 to 100 cells as well. And when we look at the patients, the overall survival of patients when they have clusters in their circulation, even in one time point during the course, they do very poorly. And so it's been a big interest in these clusters, and many groups are working on these. But one of the things we did as being engineers, developed a technology that can find these clusters specifically. I won't go too much into the detail of this, but you can imagine how we use inertial focusing that are focusing cells, and it actually focuses cells based on size. Clusters being bigger, we can actually use inertial focusing to separate clusters from as a function of size. Here you see 2-cell, 3-cell, 7-cell clusters with spike, and then isolate in blood to learn and develop this technology. And we can find these clusters now in actual patients quite efficiently. And we also use the clusters as well as the single cells to do precision oncology or medicine. Can we use these cells, because they're alive, to culture and hit them with different cocktails of drugs and see which patient is going to respond to what treatment? And efficiency is terrible. We are working on it, 6 out of 36 patients, and I'll come back to that a little bit later. But we can occasionally culture these cells and then use those cultured cells as a panel for drug screening. And in these 6 patients you see here, there are no primary in these patients anymore. Either primary is removed or they had mastectomy. So there is no way you can go and probe it again. So circulating tumor cells is your only viable way to get cells from these patients. First of all, the cells change. When we get these, there are new mutations acquired during treatment, whether it's pressure of the chemotherapy or spontaneous mutations, we don't know. But these mutations were not in the primary tumor 5 years ago. Many of these are druggable. So there are actually drugs for these mutations, but you wouldn't know if you didn't do this kind of a measurement. And so they were not present in pre-treatment tumor, but they are present now. So we could actually, I'm just showing you one example, and red being, we killed all the cells, red being, they're alive, blue meaning that they are killed. This patient had 2 of these mutations, de novo mutations, and we have antibodies for them. So this was treated different concentration of individual inhibitors versus combined inhibitor. You can see the combined inhibitor is much more effective killing these cells from the patient. We'll rarely, if any, use cocktails of treatment. So this kind of information really personalizes the treatment. We're not doing this information is from patient, but it's not actionable until bigger FDA trials done. But we do put them in animals to show that if we do treatment, if we do just vehicle treatment, no treatment, basically the patient, the tumor grows in the mouse. But if we do the combined cocktail treatment, you see that you kill the tumor, versus if you do single treatment, the tumor is still present. So you can see how you can use this as almost a living sensor to give you functional information in response to a drug treatment to figure out which drug you should use for a given patient. The big holy grail is early detection. I'll give you that example too in the next 10 minutes or so, and take questions. And typically, when you have this little coined lesion early, it's very easy to miss this. Most cancers, 95% of them, we capture at stage 3 or 4 when it is spread all over the body. The survival, if we can catch it at 25, with 2 days, not fancy, chemotherapy, radiotherapy, surgery is almost 90-95%. It's almost a cure if we can push, but 95% of all funding goes to metastatic tumor. Very little funding goes to early detection. And so we have started looking into whether or not we could use circulating tumor cells for early detection. This is for liver cancer. Of course, it's very important to do early detection because it's clinically actionable. Are you going to treat them in a certain way? Are you going to do transplantation? We were able to increase the resolution of the technology, and where we had the reason why we did the liver cancer is clinically important, plus liver hepatocytes are the only cell in the human body that has specific markers like albumin. So it's easier to distinguish these cells from non-tumor cells. And we developed this droplet PCR technique that we isolate the circulating tumor cells together with few other cells, contaminating cells, lyse it, and get the cDNA, do ddPCR, and we can detect one cell in 40 ml blood, or actually 200 billion cells. And we have done this with a number of patients. Serum AFP is the standard measurement, and then we added the CTC score. We could significantly increase the early detection from only few cases to more than 50% of the cases. The problem is it's still a tiny amount of blood. You have 5 liter blood. You can get 2 tubes, 20 milliliter blood, 0.04% of the total volume of the blood. And finding a rare event in a tiny little volume is a statistical nightmare. So the question we said for early detection, can we go to a larger volume? And so instead of 10 mil, can we actually interrogate 1 to 5 liter and of the blood volume so that we can push our resolution to early detection? To do that, we have started using a technique that's routinely used in blood banking, blood transfusion, called leukapheresis. A patient comes, and the blood is taken out, the leukapheresis device uh, spins the blood in a specific way to isolate nucleated cells and RBCs and platelets and granulocytes from each other. Then red blood cells are given back to the patient, and the nucleated mononuclear cells are taken. It's called a leukopak. And then from this leukopak, we would isolate CTCs, circulating tumor cells. This could be equivalent of 1 to 5 liter, depending on how much you do the process. So you can actually interrogate the whole blood. It will take about 2-3 hours, or you could do a shorter, softer thing for 1 liter equivalent for in an hour. And so you can measure, manipulate these parameters. So we put the white blood cell beads here, label the white blood cells, use our microchip and to isolate CTCs. The problem is this is a very complex fluid. This has 30 billion red blood cells, 180 billion platelets, 6 billion white blood cells, and only 100,000, and it's about 100 milliliter of volume. So how do we go from this to find the CTCs and reduce the volume to 2 orders of magnitude? It's quite challenging, and we have developed, when Mishra developed a chip that is the similar principle as the earlier chip, but we have brought the magnetic field right next to the channels, because we have to go to be able to process that much. We need to and still sort these white blood cells from the tumor cells with the beads that they have on them. We need much higher magnetic field. So we put a ferrofluid here that is like a lens that creates a much higher magnetic field within the channel. So we can actually move the cells in this device at 20 meters per second, but we could deflect a cell that has even a single bead on it. And I won't go into too much of the technical details, but show you some clinical results. You see that we can get this is triple negative breast cancer, liver cancer, melanoma, and we can blood volume process is equivalent of 5 liter. When it says 7 liter, it doesn't mean actually we collected 7 liter blood. In each collection, you go through a liter. You actually have to process more than that to be able to collect equivalent of it. So that terminology is a leukopak terminology. But what is you see is we can get thousands of cells. We have patients that are not in this graph, but we have had 50,000 cells. And we have also early stage patients where we find these cells in thousands and hundreds. And you can see if these cells are alive and beautiful. It is now we can push it for early detection. The power of this is that you can, as a sensor, do 2 things. I'm agnostic to the type of cancer. So somebody comes with a high-risk patient, and or somebody saw in a screening test a coin lesion in the lung or kidney, or they did there was somebody coughing. They did a low dose CT scan, which is very common now in hospitals because it's a low dose. It's used very broadly, and you see a little ditzel, a little cyst or coin lesion. You don't know if it's a cyst or it's a tumor. And so you could have high-risk patients. You could have patients from these relatively unreliable screening techniques that have a lot of false positives. And so we're asking, can we make our tool that is confirmatory. So a definitive cancer. The best way to show that a tumor cell is a tumor cell is to show for aneuploidy, single nucleotide variation, CNV. The aneuploidy, chromosomal aneuploidy, is a definite diagnosis for cancer. And here in this patient, you can see that there are variations. And so that is a definitive diagnosis of tumor. And we also showed that we could do this, in this case, precancerous pancreas, IPMN, and intraductal papillary mucus neoplasm, and you don't see those variations. So you can actually definitively show that this is a cancer cell you have. But not only that, we can then do single cell RNA sequencing, and that will give you the tissue of origin of the cell. So in this kind of a technique, you can have a perfect sensor, in a sense, that will tell you whether or not it's cancer and whether or not it's coming from a specific organ. And then, of course, you could do more detailed studies. So in this case, the way we use a broad use of this is not a screening. It's a diagnostic, definitive diagnostic of a patient. So if the patient goes to check up, they do the various screening tests, low-dose CT scans, cell-free DNA. These have a lot of false positives. If that happens, now you can't take every one of those patients and do biopsy or put them in MRI. So we position this test initially as a definitive diagnostic test to screen all these false positive, so that you can decide who should have surgery, who should have resectable, who has resectable cancer, who should be imaged further, and then do follow-up. But do this in early detection. So that's the way we are positioning initially. Ultimately, it could become a screening test, but we do not want to do that. Those clinical trials are very complicated and large. With that, let me finish. We have 10 more minutes for me to take questions, and lots of thanks to people who have done the work. And Fatih, who is a faculty at Georgia Tech, AJ, runs his own company now. Dino is a professor at UCLA, and are the people who have done the work that I presented today, as well as Sunita Nagrat, who's a professor at Michigan now, and the collaborators, Dan Haber, Shamala Maheswaran, but also Avanish Mishra, who was a postdoc now faculty, and John Ed, they are instrumental in all these studies, as well as Ravi Kapoor has done the work for scale-up of the technology, and the clinical collaborators, Lisha, David, and Rick. Is the big thanks goes to our patients. We do 3,000 to 4,000 patient sample processing every year on various projects, and we are thankful to them to believe in us and to provide under, of course, IRB and consent blood that we could study, as well as our funding sources. With that, why don't I stop? It seems like we have 10 more minutes. Maybe we could, I can take some questions.
Many thanks, Professor Tanner, for this really interesting talk. Yes, we have quite some questions in the chat.
Okay, let me open the chat. Can we filter these clusters out using dialysis? That's a very good question. And yes, we could potentially. Basically, you are saying a cluster leukapheresis or cluster apheresis system. The apheresis leukapheresis system we use to get the CTCs. Can we build a leukapheresis system that is targeted to remove clusters? Yes, we can. And we think about it. The problem with that, it could be very helpful, but it's a complicated procedure. These cells keep leaking into circulation in an active cancer. And so you can't do a dialysis every week to remove these cells. You probably need a more therapeutic approach. We have looked into specific properties of these clusters. Plaque-to-globin was overexpressed in these, and we have developed some antibodies to directly target drug target clusters and published a paper on that I didn't discuss today. That might be a more effective because dialysis is probably right now the biggest healthcare cost, the kidney dialysis, not cancer. And I don't think there's appetite to develop another dialysis system for even much larger number of people. But the idea is a very good one. We did think about it. It may not be clinically feasible, but it's a brilliant idea. And do you foresee your work being affected by the changes at NSF, NHRC, and at the government policy level? And yes, anybody who's funding is from NIH and NSF. There's concern and worry. In our case, it seems that the government policies are more related to DEI issues or funding going to those kind of issues. In our research, we're not doing that. It's cancer research, and we hope that it's not impacted with some of the headwinds we are having in the political world. But it's, we are continuing our work and we hope the best.
I'm going to ask a question myself. So what is the cost of the chip that you are using?
Yeah, the cost of the chip is actually very reasonable. It's scaled, and when it's used in larger volumes, it will even be cheaper. And right now, the plastic chip is about $30, $40 maximum. And the bigger cost is the beads, the magnetic beads that we add to blood is about per patient is about 100. So per patient, all the loaded costs of all the goods, including staining and molecular whatnot, is under $1,000. That's reasonable. And when it is really scaled and broadly used, it will come down significantly to a few $100s. So compared to if with all these false positives, the problem with these relatively lousy screening techniques is that you're going to scare a lot of people. If you go and you have a low-dose CT and that shows a coin lesion in your lung, you have two options. You don't worry, or they need to go in and do a biopsy. A lung biopsy is not an easy biopsy. It's a pretty serious biopsy. And if you do it to a large number of people, you will kill few people in the process because of infections. And some people will have serious side effects like pneumothorax and other things. And if the lesion is closer to the diaphragm, they need to do a wedge removal of the lung. So these are serious invasive procedures. Either you worry the person or you have an invasive procedure. So that's why we position this approach as a diagnosis of a screen. And there, the cost of about $1,000 is minimal compared to biopsy and imaging, that the money you will save from these invasive or expensive procedures.
There's also another question that can an mRNA therapy causing cancer self-destruction be developed from the recovered live cancer cells?
It's yes, from the circulating tumor cells. It could be done from the primary too. And the question is, of course, it's a good question. The target might be changing as the tumor is changing, as I showed in some of the slides, how it goes to epithelial mesenchymal transition. It changes as far as the resistance gene. It changes. I showed also an example of when we get them later in breast cancer, there are new mutations. So the point of the question, I take it, is that do we need different mRNA vaccines for the same patient as the tumor evolves and use these cells to do that? Yes.
have another question because you were mentioning, like, for instance, for melanoma, you were having like 49 different CTCs that you were measuring. If you want to go for like multiple diseases at the same time because you are putting it between the screening and actually the definite diagnosis, is that possible? And how many actually diseases that you can foresee that you would be able to do on the same chip?
Yeah. Well, you mean the diseases, different types of cancer, I assume. Yeah. And yes, because we do the copy number variation, CNV, aneuploidy, if you have, so we can get a sample from any person. And then you look, and if you find CNV aneuploidy in that person has tumor cells circulating in blood. It shouldn't be there. And so that's definitive. And then you do RNA sequencing of those same cells. And we could do this at single cell level. So we could get the CNV from a cell and then later do the RNA sequencing. Now you know the origin of the cancer. And so you can find the tropism, where the cancer is, as well as whether or not it's a cancer that's active, it's a tumor. And because there could be epithelial cells circulating in your blood, normal epithelial cells, because of infections, because of other type of medical conditions. And that doesn't mean that they're going to kill the patient, because it's a normal epithelial cell that's leaking into circulation because of other comorbidities. And so finding the aneuploidy is a direct evidence that it's a tumor cancer cell. And the technique is agnostic. Like I said, it's, we get rid of blood cells to find the tumor cell. So it applies to any solid tumor. We don't do it with blood tumors because there are plenty of those cells. You don't need our technology. You can do that in flow cytometry. But for solid tumors, yes, it's applicable. And you just need to, in the RNA-seq, you will get your answer.
Jerry is asking, have you applied your chip technology to early breast cancer detection? It looks like you have done some work to track CTC changes in breast cancer.
We started early detection right before COVID, and during COVID and after COVID, it's been very difficult to get patients. And so we have maybe 10 patients we've done so far in early cancers, and breast cancer is not one of them, but it's on our list. We are trying to streamline the process and primarily trying to replace leukapheresis, which is still a complicated procedure that requires blood transfusion centers. That's a bottleneck because they're so busy with patients that are for blood transfusion and CAR T-cell treatment that it's very difficult to get leukapheresis time for diagnostic purposes. But there's a different way to do that, and actually much better, much faster. There's a European technology called MicroPress, which takes a pint of blood. A pint of blood is about 500 ml, so that's 10% of the total volume of blood. Based on our calculations, that should be enough for us to do early detection. So we are going in that direction. We have small number of patients that we have successfully identified, and some pancreas cancer patient that had some major surgery that our analysis said that person should have not had that surgery, but they couldn't know. So there's very valuable information to be gained.
Thank you, Professor Tanner. It was a privilege to have you today here, and I think I learned a lot. I hope like everyone else also learned a lot, and there would be like more presentation from you in person, hopefully the next time, so that you tell us more about what you're doing, and hopefully we can collaborate on future projects together too.
Thank you, Patricia, for the invitation, and thank you all for coming. Thank you for the questions, and hopefully get to see you. And if you have any follow-up questions, you can always email me. Thank you.
Thank you. That's
Have
really
a good day.
Thank
Bye.
you.
Bye.